Another question that many people have repeatedly asked is that people now know that a virus is smaller than bacteria. So, as they know that killing bacteria is possible, they wonder if it is the same for the viruses.
As many say that to kill viruses, high concentration, such as 500 ppm or 1000 ppm, is required. I wondered on what grounds they say such a thing, and concluded that some people believed that viruses are a version of strong bacteria and a very high concentration is needed to kill them. However, as I commissioned various organizations to test, they were confirmed to be killed with 100 or 200 ppm.
You mean we don't need more than that?
I believe so. When there was an outbreak of mad cow disease, I heard a member of the National Assembly mentioning that mad cow disease bacteria cannot be killed even when boiled with 600 cc. It's quite funny because the water cannot be boiled to 600 degrees, and 600 degrees is the temperature at which organic matters just turn into air. It's a temperature that makes things disappear without any residues. If there are bacteria that do not die under such temperature, then it's something that even God cannot manage. Recently, I also heard someone saying that there are bacteria that do not die under the temperature of 100 million degrees. We are just being too ignorant. Because we are too ignorant, we say that superbacteria or superviruses cause harm to humans and they cannot be killed. But I believe these viruses are killed much more easily. When we classify bacteria, we classify them into two categories, which are general bacteria and pathogenic bacteria. Bacteria are not divided into smaller groups like Salmonella, Vibrio, and E-Coli, but largely divided into general and pathogenic bacteria. The most representative bacteria from the pathogenic category is E. coli, and there are various types of E. coli.
Isn't it big in size?
Bacteria are all big in size. They are all visible when magnified 1,000 times to 1,500. E. coli is considered as a criterion when checking for pathogenic bacteria, even for the government tests on drinkable water. By the way, at some mineral springs, the signs with descriptions of how much E. coli is incorrect. The correct way is to label it as an E. coli group. E. coli group refers to many pathogenic bacteria, including E. coli. Then why do you think we take E. coli as a criterion?
Because they are the ones that die the least?
Because they die the easiest.
Oh, because they are the ones that die the easiest.
Yes, because they die the easiest and the most. E. coli being alive is a measure that other bacteria are also alive. If E. coli is still alive, then...
I get it. Because E. coli dies easily, if E. coli is still alive, the possibility of other bacteria being alive is very high.
Of course, general bacteria do not die very easily, but E. coli does. The bacteria that cause really serious diseases in the human body actually die very easily. Some people believe that such bacteria don't die easily. And of course, there are some bacteria that structurally don't, but most of them die very easily. This is based on my research studies and opinion, though.
When comparing bacteria and viruses, which one do you think die more easily?
I believe that viruses die more easily because, as I mention all the time, viruses cannot survive alone. It's a substance that has to stick to somewhere to cause problems. That is why there are a lot of arguments among scientists whether a virus is an organism or just a substance. I view it as an organism, though, because something is created and becomes something, which is a function of a so-called seed.
Is it not viewed in that sense in the academic world?
There is a case that it is viewed as a substance in the academic world as a virus does not have a metabolic function, hence not being an organism. However, because it is a so-called seed, as it can create something, I see it as an organism.